can say that. Okay, great, off to a great start. So thanks so much, everyone, for coming. I don't want to take up too much of Rick's time, but I think everyone knows Rick, our very own Rick Karban. He's a very distinguished researcher. You name it, he's done it. He's published hundreds of papers, written several books. He's mentored many, many students, many of which have gone on to become professors of their own, and he's won numerous awards. So I'm really excited to learn more about your work. It's really an honor to have him today. So without further ado, Rick.
Thank you. Yeah, so today I'm going to talk about plant communication. And since I'll no doubt forget, I should right at the start acknowledge the people who have really contributed quite a bit. Hey, come back. Have really contributed quite a bit to this work. Harry and James. Harry's done a bunch of the field work as well as the chemistry. James has also done some field work, but mostly chemistry. Satomi has done whatever genetics that we'll talk about. She has done that, and Patrick has done lots of the field work. So today I'm going to try to address these 7 questions. And the first 4, what is communication? Do plants actually communicate about herbivores? Why might plants respond to cues? And are these cues public or private information? And these are all kind of, at least from my point of view, like a little bit old hat. So I'd like to go through them really quickly. But in doing so, I might well use jargon or make assumptions that you can read my mind and have already heard this talk. So please interrupt me as I go through these 4 pretty quickly. The last 3 are the ones that are more recent and more interesting to me, and I'd like to spend sort of more time talking about them so that I am forced to sort of flesh out my own ideas, and perhaps you'll have questions about them. Do these cues, is there genetic variation in these cues? Is there geographic variation in these cues? Have the cues perhaps converged to a single cue or very few cues when the risk of getting eaten is high? And then finally, I've been really influenced by the many people here who have done fantastic work demonstrating both that animals have personalities and that those personalities might be important. So I'd like to do a little bit of the same thing for my plants and ask, do these plants have personalities? And as I say, I'd like to spend most of my time with these last 3 questions. So first, let me tell you what I mean by communication. When I use this term, I'm talking about cues that are emitted by a sender that can be perceived by a receiver. And when the receiver gets these cues, they cause rapid and conditional responses in the receiver. This is certainly not the only definition of communication. Many people in animal behavior who have been thinking about this stuff for much longer think that when they use the term, they want it specifically to apply to instances where the sender intends to emit the cue. And intention is very difficult to determine for animals, for people, let alone for animals, and it's kind of ridiculous for plants. I recognize that, and I recognize that what I will be talking about, many people would rather have me describe as eavesdropping. But nonetheless, I will use both of those terms. And this is what I mean, that a cue is emitted when a plant gets attacked by its herbivores, and other tissues on that same plant, other neighboring plants can perceive that cue and respond to it. So I'm particularly interested in the possibility that plants are communicating or eavesdropping on one another with regard to damage by herbivores. And I hope that I convince you that this actually happens. And this is kind of a cartoon of the basic experiment that I've done many times now. I've got pairs of sagebrush plants out in the field, and I have 3 treatments, if you will. In this top treatment, I experimentally clip the leaves of one of these plants, and then I assay the amount of damage in a neighboring plant. So I'm not messing with the assay plant, but the neighboring plant is where I'm measuring damage. So I call this a clipped treatment. And then I have plants that are clipped, but then I immediately put a plastic bag around them to reduce the release of these volatile cues. This is clipped and bagged. And again, I'm assaying a neighboring leaves on a neighboring branch. And then I have a control that never gets clipped. When I do this, or when we do this, these are the kind of results that we get. And so in this graph, and in most of the figures that I'll show you, the y-axis is the same. It is the damage to the assay branch, so not the branch that I experimentally clipped, but the neighboring branch, and how many of the leaves, what percentage of those leaves get then attacked by chewing herbivores over the growing season. And I do my experimental clipping at the beginning of the growing season, as soon as the snow melts. Okay, so what you can see is that the plants that were near to a plant that I experimentally clipped with scissors has fewer damaged leaves than the control. And when we prevented the volatile cues from moving between the experimentally clipped plant and its neighbors, those plants also got relatively a lot of damage. So this suggests that there is some kind of volatile communication. We've done other experiments. I don't remember if I've got a slide of this, where we just move the volatile cue, and we move it fairly great distances. So the volatile cue is sufficient to provide this decrease in damage over the season. So a volatile cue seems to be both sufficient and seems to be required for this kind of communication. There might well be communication through soil-borne channels. We've not seen that in this particular system. Okay. Something about sagebrush plants. These plants were close together. I chose pairs of plants that were close together when we did these experiments. And sagebrush plants are quite often neighbors of one another. This is on the left is just a photo of one of my field sites, and it's quite dominated by sagebrush. Sagebrush is a really common plant throughout the Great Basin. We can document communication occurring up to a distance of 60 centimeters. And it's just as strong, really close, as it is at 60 centimeters. At 60 centimeters or beyond 60 centimeters, we fail to detect an effect at all. And, you know, presumably this will be affected by wind speed and all this kind of thing. But it's when we have measured this, it's quite a kind of plateau, quite a steep curve. We find that young plants are the best emitters and receivers. And in the cartoon that I showed you, we did the initial experimental treatment using scissors. We also used real herbivores, caged beetles or grasshoppers on some plants, allowed them to do the damage, and we get much the same kind of result. Doing that is a lot more work. So for the vast majority of the experiments that we've done since then, we've used scissors to do the clipping. Why might plants respond to these volatile cues? For one thing, many plants, sagebrush included, are not very good at sort of coordinating their own defenses through vascular connections. Some plants certainly can do this to some extent. Some plants, when you damage one leaf on one branch, without volatiles, they manage to induce systemic resistance. But quite a few plants are incapable of this or very poor at it, particularly plants that grow in arid regions such as sagebrush. We've done a similar kind of experiment in which we, again, have 3 treatments, but instead of using pairs of plants, we have done this just on different branches of the same plant. So here we've clipped this branch, and then we assay damage on the neighboring branch. We did the same thing, but we, after clipping, stuck a plastic bag around the branch to reduce the possibility of volatiles moving from one branch to the next. There's still a possibility of vascular communication, and then we have our control. And what we found is similar to the situation where there were 2 bushes. It appears that volatile communication is pretty much required, at least under these conditions, is required for the plant to coordinate its own systemic defenses. And I think it is reasonable to suspect that perhaps the communication between individuals that is probably more exciting to me, to most of the people in this field, that might have evolved as after communication that is required to coordinate an individual's defenses. Okay, so this communication is required just for systemic induced resistance. In most years, there's very little recruitment of sagebrush. Sagebrush is a slow-growing, long-lived shrub. I've now seen in about 30 years worth of working on this system, I've seen recruitment events 4 times. And 2 of those times were in 2010 and 2011. And so in both of those years, we decided, okay, let's sort of drop everything and really take advantage of this weirdness. Sagebrush is actually reproducing. And so we marked a bunch of plants, and we looked at the survival of these seedlings over a single growing season when the seedlings were close to clipped adult neighbors or unclipped adult neighbors. And what we found in both of these years is that the vast majority of the seedlings died over their first summer, but that about an order of magnitude, survival was like an order of magnitude better when those seedlings were next to adults that we had experimentally clipped. So maybe this sort of eavesdropping is associated with increased survival for seedlings. When we looked at how well adult plants fared next to clipped or unclipped neighbors, we found that plants that grew next to clipped neighbors produced more new branches and also more inflorescences. So that these are pretty terrible proxies of fitness, but nonetheless, proxies of fitness are hard to get for a plant like sagebrush. And they suggest that there probably are advantages to eavesdropping on your neighbors, to being able to eavesdrop on your neighbors, even when the cues are of questionable validity, because to be extremely anthropomorphic in clipping plants and setting these things up, we are essentially giving them the cues that there's a ton of herbivory, that herbivores are completely trashing their neighbors, which wasn't really the case nonetheless. This is definitely worrisome for me. Nonetheless, we see these benefits as well as we can measure them of being good at eavesdropping. Is communication public, or does it use private channels? And the answer to this question is both. There's evidence for both kinds of cues. One of the first sort of convincing examples of communication was this paper by Ted Farmer and Bud Ryan. Bud Ryan's lab had worked forever on the chemical responses of tomato plants and in Washington State, at Washington State University. So in the sagebrush region. And somehow Ryan knew that sagebrush was a mega producer of methyl jasmonate. And this was before methyl jasmonate was recognized as an important plant hormone. So they did this experiment where they incubated tomato plants with a cut sagebrush branch or tomato plants without sagebrush, and then measured putative defenses in those tomato plants, as well as how well herbivores did on those tomato plants. And what they found was that the tomato plants that were incubated in the lab in vacuum jars with sagebrush plants, and these two species never occur in nature together. Nonetheless, the tomato plants showed elevated levels of things that they assumed were defenses, and also weren't as I forget if they did a performance assay for caterpillars or a behavioral assay, but the caterpillars didn't like the tomato plants as well. So some of these cues are really general. Some of the cues are really specific. We did an experiment in which we cloned a bunch of these sagebrush plants, took cuttings. Cloning sounds like high tech. We made cuttings of sagebrush plants, grew them up in the greenhouse, and then moved them back to the field so that we're able to look at communication between tissues that are either genetically identical, that are cuttings of the parent, or communication between individuals that are genetically different. And what we found is that this made a really big difference. So in this experiment, we have 4 treatments, an unclipped control. They got lots of damage. A a treatment where the assay branch, the assay plant itself was clipped. That got a lot less damage, but the interesting ones are here. When the Q came from genetically identical neighbor, the reduction in damage was greater than when it came from a genetically distinct neighbor. So plants seem to be able to communicate more effectively when the communication is between, essentially with themselves. And this got us wondering if relatives communicated more effectively. Satomi developed a system of microsatellites, and she was able to tell us the relatedness of all the individuals in our meadow. And then we moved the cues around and did this using this super syringe. Yeah, damaged plants, let the cues accumulate in a plastic bag, got the cue inside the super syringe, walked it to another plant, and incubated that plant with the cue. And we did this in a lot of experiments that were just a little bit different, but what we consistently found was that when the cue came from a close relative, damage was reduced compared to when the cue came from essentially a stranger, a distant relative. Okay, so there appears to be a genetic component, and this suggests that perhaps communication is mostly with self, with tissues that are on the same plant, or with kin. And I should say that the sagebrush is strongly genetically structured, so that individuals that are growing next to each other tend to be quite closely related. So this makes it less likely that an emitter will aid strangers, much more likely that plants are responding to themselves or to their kin. It made us wonder whether or not kin share susceptibilities. And this sounds like it would be a pretty straightforward thing to measure, but we get sort of different answers to this in different years. So maybe, and it also got us interested in thinking about what the active cues might be, or sort of what is the language that plants are using. And that's largely what the next little bit of the talk will be about. Oh, okay, here we go. So when James has collected these volatiles and analyzed them with GCMS, he is able to find a lot of peaks, depending upon sort of the fanciness of the machines and the fanciness of the techniques. People have measured about 100 volatile peaks that are emitted by sagebrush. So the potential language is really enormous, and sort of, we're never gonna get to the bottom of this. James also noticed that the volatile profiles of different individuals tended to be kind of organized into a few repeating patterns. So each of these pie diagrams represents an individual sagebrush plant in the same meadow at Sage Hen. And the spatial orientation of these pies is just how they wound up. They are not organized in the meadow in this way. Just we move the pies around, and you can see that there are two common sort of pie, whatever these would be. There are two ways they look. They are dominated by this apricot color, which is thujone, or they are dominated by this kind of, what should we call that? Raspberry-like color of camphor. And most individuals have one of these two dominant chemicals, as well as the same minor peaks. There are a few individuals in the meadow that are kind of oddballs, that are a little bit different than these common chemotypes. And we will come back to these. So it appears as if these cues are organized into repeating what other people and we have started to call chemotypes. And when we have moved volatiles between chemotypes, we found that the communication is more effective between individuals of the same chemotype relative to different chemotypes. When we've looked at parent offspring regressions, we find that these chemotypes are highly heritable. This is strange. So Patrick has now looked at the volatile emissions of, oh, here we go, of more of these chemotypes or more plants over a much larger geographic area and found additional chemotypes. When we have looked at the chemotypes sort of up and down the east side of the Sierras, the green on that map shows the range of sagebrush on the east side. What you can notice from these pies, so each pie is a different site on the east side. And some of the sites are really dominated by one or two chemotypes. This gray and pink are camphor and thujone, the same ones that were at Sage Hen. Oh, Sage Hen's in the upper right. Mount Rose is dominated by a different chemotype. There are other sites that have a lot more variation. This is Valentine Reserve. It's got, I don't know, 6, 7, 8 different chemotypes that are pretty common. And looking at this, darn. Ooh, that's not good. Okay, looking at this, what we noticed was that at those sites that get a lot of damage, so those are the sites, this is the kind of proportion of leaves, the amount of damage on the x-axis. Those sites that get a lot of damage have lower chemotypic diversity. The sites that have lots of different chemotypes are the sites that don't get a lot of damage. This is kind of interesting and has really been the motivator for what we've been up to the the last few years, we've tried to ask whether or not cues have converged in response to high levels of herbivory. And I'll explain this in a little bit more detail. And one reason that we suspected that this might be the case is because bird songs, alarm calls of birds, apparently show this kind of pattern. So in those situations where predation risk is really high, the alarm calls of a bunch of different bird species have converged so that they can all understand each other's alarm calls. So apparently the tufted titmouse on the top is, it has the best view of predators, and it's also kind of the biggest talker. And the brown creeper, the woodpecker, and other species that co-occur with the titmouse in situations where risk of predation is high, have all evolved to be able to understand and respond to the alarm calls of the titmouse. When predation risk is not high, this has not occurred. They don't respond to the alarm calls of the titmouse. And an experiment that Einokowski and other people in Andre Kessler's lab did with goldenrod also suggests that this might happen for plants. So they have a situation where they have plots of goldenrod that they have excluded herbivores from. So some of these plots have not had any herbivores for 12 years, I think. And others get really high rates of herbivory by beetles. And what they found is that plots that had high rates of herbivory, where they allowed the beetles to do their thing, had open communication and convergent cues. Everybody could respond to everybody else. Where they protected the plants from herbivory for 12 years, they had divergent cues. They had a lot of diversity in the cues that the plants were using. So this is a, I think, really cool selection experiment. It's done in one place. Yeah, selection experiments are always elegant to me. And it made us wonder if something like this could be responsible for the correlation that we see. So the correlation that we have seen on the east side of the Sierras is that there's a relationship between chemotypic diversity and levels of herbivory at those sites that we know have gotten sort of chronically high levels of herbivory, just because we've been there over the past number of years. High herbivory is associated with few chemotypes, and not much herbivory is associated with more chemotypic diversity. Okay, this correlation can be driven in either direction. So it could be that where herbivory is high, there's low chemotypic diversity, because low diversity allows for effective defense, so that rare chemotypes should suffer. Rare chemotypes should get eaten more and be selected against and be less common and be, well, yeah, be even, yeah, less common. It's also possible that causality goes in the opposite direction. So lots of people have found that in situations where there's low chemotypic diversity, phenotypic diversity, that herbivores do really well. So think of sort of the potato famine in Ireland, where you have, everything is genetically identical, it's really a great situation for those pests that are able to exploit that particular set of defenses, and they do really well. So it's possible in this situation that that rare individuals would actually do better. And so we've looked at, we've sort of gone out of our way to look at a bunch of populations that are either at Sagehen near Mount Rose or down near Mammoth Lakes where Sorrel and Valentine are, and mapped at a fairly fine scale the chemotypic diversity of these populations. And it kind of, so we've got about 30 of these, and these are just sort of a sample. This was from the first year that we happened to do this. At Mount Rose, levels of damage are really high, and the rare chemotypes really do not do well. At the other sites, levels of damage are much lower, and the rare chemotypes are fine. And so we've now wondered if this same pattern would apply more generally, and if this pattern might provide some evidence about the kind of selective drivers of the communication system. And so for a bunch of different populations, we attempted to calculate the advantage of common chemotypes. So this is simply how many leaves of the rare chemotypes get damaged minus how many leaves of the common chemotypes get damaged at that same site, divided by the mean level of damage. So if the rare chemotypes are getting a lot of damage, then this number is positive, and there's an advantage to being common, to being like the common chemotype. If the rare chemotypes don't get much damage, then this whole number is negative, and there's a disadvantage to being common. And so we have looked at this for a number of different sites, and we do see a positive relationship between level of damage and how well rare chemotypes do. So at places where there's lots of damage, it's good to be able to, it's good to be of the common chemotype, presumably because that allows you to eavesdrop on your neighbors and gear up your defenses when they need to be geared up. There's no particular advantage to being of the common chemotype when herbivory is not very likely. So that's consistent with the idea that the convergence of chemotypes might be favored when rates of herbivory are really high. At some of these sites, at Sagehen, these SH sites, and Mount Rose, we've gone back in a bunch of different years and measured the advantage to common chemotypes, and we find much the same kind of pattern that in those years when there is a lot of herbivory, it's good to be of the common chemotype. In those years when there's less herbivory, it doesn't matter, maybe it's even disadvantageous to be like everybody else. And so I think the argument that I am trying to make, that I'm interested in seeing if this flies with this group is that herbivory is driving chemotypic diversity because low diversity allows plants to effectively communicate with their neighbors, to eavesdrop on their neighbors. And rare chemotypes will suffer and be selected against. Yeah, so I'm interested in hearing what you think about that. Let me quickly tell you a little bit about attempts to see if plants have a personality. And what I mean by that is that there's a behavioral tendency that is consistent either over time or not either, but over time and across different situations. And I'm just sort of borrowing that from the definitions that animal behaviorists have thought a lot about and developed. And in order to ask that question, what we've done is moved cues from a single emitter and given those cues to a bunch of different receivers. And we've done the same thing for a bunch of for sort of in reverse. So we have actually in this cartoon just 5 different emitters and 5 different receivers. In fact, we've done it for each plant with 10 emitters and 10 receivers. And what we find is that individual receivers, regardless of where they are getting the cue from, some receivers don't get almost any damage. Some receivers get a lot of damage. This isn't very surprising. Where the plant happens to be will strongly influence how much damage it gets, its environmental conditions and a million other things. What is perhaps, I think, more interesting is that there's consistent variation, albeit not as much, among individual emitters. So some plants are really effective at providing cues that other individuals respond to. Yeah, this seems a bit more interesting and surprising. Good communicators in one year are also good communicators in the next year. And those individuals that are good receivers also tend to be good emitters. So as time changes or as situations change, individuals that are good communicators remain consistent. So the take-home messages are, I hope I've convinced you that plants actually do communicate, or at least eavesdrop if you prefer, to reduce losses to herbivores. That communication is more effective among tissues that are genetically identical among kin, among individuals of similar chemotypes than they are among those that are more different. That high risk may have selected for a convergence of chemotypes so that individuals can more effectively respond to the cues of their neighbors. And then finally, and I'm sorry I rushed through this, that individual plants might have personalities with respect to neural impulse. So I'd be very happy to hear what sort of comments people have and what you think. Thank you. Yes.
That was a really awesome talk and super cool. I was just curious that a lot of times in literature, when plants are releasing those volatiles like parasitoids and other natural enemies of the pests tend to listen in. Do you find in areas that are more or less diverse with chemotypes, is there a higher, lower, or different diversity of these natural enemies?
Yeah, that is a good question. I have been someone who's thought that the whole business of these cues attracting natural enemies didn't apply in my system when I was mostly focused on grasshoppers. I did some experiments to see if plants that got damaged recruited more predators, and they did, but those predators didn't have any effect on the grasshoppers. So I sort of poo-pooed the idea. More recently, I have found that beetles are grasshoppers are always present. They always take, I don't know, like 3 to 10% of the foliage. Beetles are much more ephemeral, much spottier, but they can do much more damage. And predators have much more of an effect on the beetles. So I've not circled back to ask any of those questions about predators, but I think they'd be really good questions.
Awesome, thank you.
Yeah.
May have missed the answer to this question,
That's okay.
buried in the talk somewhere,
Yeah.
but. So I get your idea there, number 3 on the list is cool, but what about the other side of it? What is it that promotes sort of high diversity? Because
Yeah,
it seems like that there's a tension there.
yeah. I don't really know, so I will wave my arms a bit. And I think that if herbivory is not a big selective force, that being just like everybody else, and I'm assuming that chemotypic diversity is somehow correlated with other traits, so that if you are just like everybody else, you are suffering more competition, and that if herbivory is not a big problem, then being different from everybody else might actually be favored. I don't know that that's true, but that's kind of the arm waving argument that goes on in my head. Yeah, Rachel.
Can I ask a follow-up on that one? I mean, this strikes me as sort of the opposite take of how people explain chemical diversity in the tropics, right? Because there, isn't it that trees that are co-occurring are less similar chemically than you expect by chance? And that is also explained by the presence of these herbivores, or people, cite the presence of herbivores. So it strikes me as like kind of a different interpretation. And I wonder if herbivore specificity or, generalist versus specialist could play into this somehow.
Yeah. So this is certainly, the kind of opposite of the Jansen Connell kind of stuff. Yeah, I don't know. It's what we found. And the beetles that are the really big, the things that are causing the big damage. So that that relationship is really driven by the points in the upper right. And there are like 3 or 4 of them. If I get rid of, Mount Rose, it's the one that is always trashed every single week. If I get rid of that, I still see the positive relationship. Yeah, I don't know.
It's cool.
Yes.
Super cool talk. I like all information and kind of thinking about that kind of stuff. In the fish literature, there's this idea of the threat sensitive hypothesis, where the higher the concentration of the threat, the more behavioral response the animal will have. So I'm wondering if you have any sort of data that shows the closer the plants are to the clipped focal individual, the stronger the response they have, and how does that volatile chemical travel across space? That sense.
Yeah, I mean, that is a great question, and certainly what I would expect to see, but we didn't. Like, we found that up to about 60 centimeters, so like yay far apart, there was a strong response. Beyond that, there was not a strong response. Yeah, I was expecting like much more of a greater response, and how these cues are moving in the environment, ooh, I don't have any idea, and my guess is it's not a simple answer. Yes.
So assuming that persistent high herbivory eventually reduces reproductive fitness, it seems like there'll be selection towards higher levels of self-pollination over time. Do you see any evidence of that? Have you looked at that? I don't know if sagebrush is
I
amenable.
don't know. So like at the Mount Rose site, for, I don't know, like it's probably been a decade. The plants have gotten defoliated over and over and over again. And sagebrush is not very tolerant of herbivory in general. And many of those plants are now dead. I have not looked at levels of selfing, outcrossing. I like that idea, but I don't have a clue. Yes.
What's the response that the receiver is doing that causes the drop off? Are there particular chemicals that go up that deter herbivory?
Yeah, so there are, yeah, like gene products that go up that we assume are important. And I don't even remember. I am so not a chemist. I don't
But it's a
even remember.
subset of the,
It's
hundred
a small
piece.
subset.
right, yeah.
Oh, and then some of these peaks might themselves be deterrent. So those, the things that I can't remember are like gene products that we imagine are like part of the defensive production of the plant. But it is quite possible that some of the actual volatiles are themselves repellents. And there is a little bit of evidence that that might be true. And that was actually something that I was thinking about looking at this summer. It is, Louis actually had the exact same suggestions. I think I might do that, take a look at that. Yes.
Have you considered memory, like how long after the initial cue does this resistance stay in the plant?
Yeah, we have done a little bit of that. So the, I glossed over the details of this. This really only seems to happen if the cue is delivered very early in the season. So the plants have a fairly short window of being responsive to the cues. The plants are doing most of their growing during immediately following snow melt. And that's when they are responsive to these cues. But that seems to put into motion changes that persist. There are 2 periods of time when the plants get a lot of damage, either early in the season when they're growing a lot, and then at the end of the season when all of the grasses and the annuals are gone. Then the things return to sagebrush and eat it again. And the resistance lasts at least until then. It doesn't seem to, well, I can't say that. We haven't detected an effect in the next growing season. Maybe there's one, but it's smaller, less easy to detect. If it's not a really big effect, we're gonna miss it with these assays. We've also been interested in knowing how long the queue is active for. So for instance, if a plant is just given a quick pop of these volatiles, it doesn't respond. It needs to have an exposure of 24 hours in order to do whatever it's going to do.
Well, thank you everyone for coming and thank you. Rick.
Yeah. Thanks.